Hi, glad to be here to introduce our deep learning works for the breast cancer recognition. And first, there are some backgrounds and motivations for our projects. To have a big picture of our work, eventually we want to predict the cancerous region of a slide. Here, there are some model like UNET that already did the region thing. It directly predict the region. But for our work, what we did is to split our slides into very small parts and do a binary classification of each small part. Use different color to label them, and at the end, we map all those labeled small part together to map them to form an entire slides, entire picture, such that we can also achieve our goal to predict the cancerous region. And in terms of our work, first we want to talk about the data processing part. Here are some issues with the data. First is the class imbalance. We naturally have more healthy pixels than cancerous pixels in each slide. So when we do the patch extracting, we will naturally have more negative patches than those positive ones. So later we will have some techniques to deal with this problem. And the second issue is about the data size. With a high resolution level, the data can be very large. And also for each slides, for each TIFF files, it's about more than 1 gigabytes. That can be a very big data set that use a lot of memory and can also cause many obstacles during all the processing and training things. And also there can come with many patches after we splitting each slides. So to deal with the problem mentioned before, we did some data splitting. The first about the magnification level, we only focus on level 2 and level 3 data. So the relatively high resolution can support a relatively better model performance. And to avoid a too large data set, we just drop the level 0 and level 1. And each patch and the patches are divided by level. They're separately stored in level folder, and each model only trained on one level. We also save our patches on the corresponding labels while we extracting them. We store them to the Google Cloud, so we won't bother to process them over and over again to process such a huge data set over and over again. And in terms of the train validation test data set, there are 17 slides splitted, processed for train and validation. We just shuffle them and take 20% for the validation. And there are 3 slides excluded for later testing. About how we determine whether the patch is positive or negative, we just look at the mask. If there is something in the mask, we say it is a positive patch. But if there's nothing in the mask, first we want to be sure that this region, like this sub picture, is for the tissue, is for the flash. It is not for like something else. So if there's nothing in the mask and there's more than 20% region of tissue, we say this is a negative patch. In order to balance the data, like to add more positive patches in the data, we repeatedly search the slides to extract more cancerous examples. So, but this will definitely cause duplication of our positive sample. So we did some data augmentations to deal with this problem. For our data augmentation, we just do some rotations, brightness, saturation, hue, and the contrast. Very similar to what those people did in the paper mentioned in the class. And here it's about all the data processing thing we do. So next we will move on to the model part.
So in this project, we use a transfer learning, and we use the pre-trained Inception v3 architecture, pre-trained on ImageNet images, and we added an additional dense layer after the pre-trained model, and we also applied an output layer which uses sigmoid function so that we can predict whether a slide is cancerous or not. And we did the same modeling for both level 2 and level 3 images. After testing and been trying for 20 epochs and 10 epochs, and we eventually decided to use 10 epochs because the validation accuracy seems to be at a constant level after 10 epochs. And we used a batch size of 16 for both models. After that, I'm going to introduce the result that we have from the previous two models. We used accuracy as our metric for both models, and on this slide we have the accuracy of training and validation datasets for level 2 and 3 images. And on the left we have the model for Zoom level 2, which attained the highest validation accuracy of 86%. And on the right, we have the model for level 3 images, which reached the highest validation accuracy of 81.6%, so around 82. And obviously, the model that was trained on Zoom level 2 image datasets attained a better result than the one that was trained on level 3 because, well, Zoom level 2 images do have much more details for the model to learn from. So the result is not surprising to us. As a visualization of our testing images, which we have two, and we illustrated the ground truth slides and the prediction slides. We have two images in our testing dataset, and this is an example. This is one of them. On the left, we have the original slice on the bottom, and on the top, we overlaid the mask that indicates the region of interest, which are the cancerous cells. So we can see that on the right, the heat map is composed of small slides, and each slide is given a probability or confidence level of whether or not it's a cancerous cell. So that probability is indicated by the color of the slide. So we can see that the most probable areas of cancer cells are in dark red, which actually can correspond to the original image. So we can see that the model is actually doing a decent job of identifying the area of the cancer cells, although there are several noises that are also in red that might interfere with people's identification of the cells. Next, I'm going to introduce some further researches that can be done to improve this project. For future endeavors, one of the major obstacles that we encountered in this project is actually limited resources to RAM and disk space. And given the dataset is massive, and we also have a pretty large model, we often encountered constant crashing of our notebooks. And despite that, we actually tried to save checkpoints and save models and save pre-processed images, but it still takes quite a long time to reconnect to the runtime every time it crashes. We actually tried several ways of producing the heat map, but this is the only way that actually runs that without crashing. So if we can have some improvements on the hardware, we can train our models with probably Zoom level 1 images and to have a larger and more complex model, which could all be promising in achieving a better results. Secondly, we can also try the UNet model, which has proven to be critical to the biomedical imaging area. While we're using sliding windows to crop out images, UNet can be as detailed as down to pixels. So UNet model would be much better in producing a visualization of our results and could be more clear to medical people who would use this model to identify cancerous cells. And lastly, we can try to customize more metrics to evaluate the performance of our model or other further models, so that it helps us understand the performance more thoroughly. This is all, and thank you for watching.